In this video, we're going to examine the proteins, namely the spike protein and its ACE2 receptor, and see how that interaction leads to the initiation of the infection process. So you're familiar with this model of the virus, but there's a lot of color going on here. So I'm actually going to substitute this really complicated model of the virus for this simplified model of the virus, in which we have these red spike proteins in a gray lipid membrane now, with a blue RNA genome inside. And you can see here that this virus has docked onto a patch of cell membrane with an ACE2 receptor protein embedded in it. So the spike protein recognizes ACE2 and binds. Okay? And that's that first critical interaction between the virus and the cell. Remember, this is happening in your lungs. And that first cell that lines your respiratory epithelium. And then this is going to trigger the receptor-mediated endocytosis that we talked about just a few minutes ago. So let's look more specifically then at the structure of the spike protein and the structure of this green ACE2 protein. All right, the spike protein, I have a model here of the spike protein. So it's a homotrimer, just like the hemagglutinin. In two of the subunits, we have colored, we've represented as what we call a surface representation. So we've got a purple subunit, we've got kind of a light magenta subunit. And then the third subunit, we've colored blue, but instead of representing it as a surface, it's what we call an alpha carbon backbone model. And what we mean by that is that every place there's a bend here, every place there's an elbow in this structure, that represents an alpha carbon of an amino acid. And we're just joining one alpha carbon to the next alpha carbon to the next. And in doing that, you can sort of trace the path of the protein in these various domains, from the N-terminal end, which is over here somewhere, to the C-terminal end, which is down here somewhere. Okay, and we have two shades of blue because we want to focus on the part of this spike protein that binds to the ACE2 receptor. And the part that we want to focus on is this right up here. This is the receptor binding domain. And the other thing that adds to the complexity of this structure is that there's only one receptor binding domain that has popped up into the up or open conformation. The other two are still down lower in a closed conformation here and here. So the interesting thing is when the structure of these proteins are determined, very often you will find this arrangement in which one of the receptor domains is up, receptor binding domains is up, and the other two are down. So what we want to focus on then is what's special about this receptor binding domain that allows it to recognize and bind to its receptor, the ACE2 protein. So we have a close-up of that interaction. So let me put this down right here and bring this model in. Here we have the green ACE2 protein. This is the extracellular domain of that ACE2 protein. Functions as the receptor for this spike protein. And here we have a slightly scaled up version of this receptor binding domain here. And what you can see, and perhaps here, there's the specific interaction between receptor binding domain of the spike protein and a particular surface of the ACE2 protein. Now, what we've done here is, again, these are alpha carbon backbone representations of these two different proteins. So we don't show any of the amino acid side chains except for 10 amino acid side chains that are on this interacting surface of ACE2. And then I think there are 9 amino acid side chains on the receptor binding domain. And we show those because those set up specific hydrogen bonding or electrostatic interactions between the two proteins. And let me stop and say right away that this interaction is somewhat unusual because in general, any two proteins that interact, in general, they interact through hydrophobic domains. So it's somewhat surprising, to me at least, that here we have a very specific protein-protein interaction that is almost entirely polar as opposed to nonpolar or hydrophobic. This is an acidic side chain. This must be an aspartic acid. And if you know your amino acid side chains and their chemical properties, you would say, aha, that's a carboxylic acid group. That's negatively charged. All right, when I bring the interacting domain up here, you see that there is a lysine side chain coming in from the spike receptor binding domain. And lysine is a basic amino acid that is positively charged. So right there you can see what we call a salt bridge. This is an electrostatic interaction between a negatively charged aspartic acid, positively charged lysine. So that's just one example of the principles of chemistry that stabilize this interaction. So there are two things going on. The overall three-dimensional shape has to be such that these side chains can come into close enough proximity that they can interact via hydrogen bonding and electrostatic interactions to create this binding. So that's the molecular details. That's the structural biology behind how a receptor binding domain on a spike protein can recognize and bind to a part of the surface of the ACE2 protein embedded in a cell membrane. This is what triggers or initiates the first step, the next step in the infection process, which is that receptor-mediated endocytosis. Okay, maybe just to summarize this whole thing again with models, you've got a virus, coronavirus here with spike proteins on its surface. You've got a very small patch of a cellular membrane here with a green ACE2 receptor protein. This virus will be floating around and it will find and bind to the ACE2 protein. And then in the process that we talked about with David Goodsel's molecular landscape, we saw how there's a pH-induced conformational change in this spike protein, which brings these two membranes closer and closer together. And when the membranes fuse, that creates actually a hole through which the viral RNA can now go to get into the cytoplasm of the cell that we're trying to infect. That's the infection process.
Hi there, Mark here, a colleague of Dr. Tim Herman's. And if you enjoyed this video, make sure to check out our other coronavirus resources available at www.3dmoleculardesigns.com/scienceofcoronaviruses.htm, including a paper modeling activity where you can create your own physical model of a coronavirus. We hope you enjoy, and thanks.